There are lots of other reasons for culturing different species, and I'll try and give you some of the main reasons because I don't have all day today. So we can go into production and preservation of animal genomes. So we can have elite breeds, rare breeds, sterile animals, diseased animals. We can clone companion animals, or we can go for genetic modification, which is, as I say, one of the reasons we wanted to do this, and there are a bunch of reasons, biopharmaceuticals, nutraceuticals. I'll try and go through a few examples of these in the time allotted. So, this was a high genetic merit cow in New Zealand who was too old to breed, and so they cloned her. So these are all clones of this cow, Elizabeth. So therefore, that's preserving the high-quality genetics of this dairy cow. This is a thing called the Enderby Island cow, and I won't go into this, but this is basically some cows were taken to an island off the South Island of New Zealand and then abandoned there. And they spent 20 generations of inbreeding. And at that time, New Zealand government decided they were destroying the habitat, so they sent the army in and killed them all. But they saved one female and took her back to New Zealand, but unfortunately they couldn't get her pregnant. Eventually they made a test tube baby of her, but at the same time they actually cloned her, and this is a copy of her here. So this is preserving a genome which had modified itself through to inbreeding when abandoned on. So it's an interesting way of preserving those genetics. Cloning, there are lots of wild cats in populations in zoos, some of which have diseases and some of which are just too old to reproduce, and actually getting those animals from the wild is just not possible anymore. So cloning allows the diseased animals and the old animals to be brought back to a young stage, effectively, to maintain the colony. Horses, most horses are gelded, so they're sterile. So if you then want to breed your horse when it becomes a champion, you're unable. But if you clone it, you've got the same genetics, so you can breed that animal. The possibility is with some rare breeds, this is a mouflon and this is a galah. These were cloned using eggs from very closely related species. Unfortunately, both of these animals died, probably because the egg was not compatible, and there were probably differences in the egg. Some people have suggested we can clone companion animals. This is CC, and this is CC's clone. Well, this is the clone, and this is the animal, but they're different colours. And this is to do with epigenetics and how development in utero occurs at random, and so they actually come out as different colours. The dog actually looks the same. This is Snuppy, the first dog that was cloned in Korea. But there are problems. In fact, they may not be identical. Now, one of the reasons for cloning was to get genetic modification. Why do you say you need cloning for that? Well, because the objective of genetic modification is to produce a stable and heritable change in the genome of an animal. Well, not only do we want to add genes, we'd like to be able to remove them or to modify them. So you may say, well, why? Well, with nuclear transfer, this gives us a way of doing this because we can grow millions and millions of cells in the laboratory, and we can apply molecular biology techniques to modify those and select them, and then we can make an animal from one cell, and we can produce an animal which is basically identical to the genetic modification or contains the genetic modification in that cell, and it will transmit that genetic modification to its offspring. So as I said, what are the uses? Well, biopharmaceuticals, produce of human proteins which are found in people with genetic deficiencies. So people with cystic fibrosis, for instance, people with haemophilia, we may be able to produce those proteins in a clean manner which doesn't require using human blood, so we have no problems with infection, with hepatitis, HIV. It's cheaper. Nutraceuticals is just making animal products more healthy, better for you, low-fat milk or something like this. The idea of xenotransplantation, the idea of using animal organs for transplantation to humans. We might want to alter production traits, so we might want to change the amount of fat in the tissue of an animal. We might want to make animals resistant to disease, or we may want to use them as models of human disease because the mouse is not a good model for many human diseases. So just a few examples, I'm not going to go through them. Clotting factor for haemophiliacs, fibrinogen for tissue glue. These are just some of the things that we were making in transgenic animals. And just to show you some pictures that we can actually achieve this is Polly. She was the first transgenic cloned lamb, born in 1997. And because, obviously, if you want to make a protein in the milk of an animal, you need to get more than one. So you can either breed them for a while, which takes a time, or you can just make some more to accelerate the process. So this is Polly and her clones. These are just some other animals we produce. These produce an enzyme called extracellular superoxide dismutase, which may be useful for preventing free radical oxygen damage to organs for human transplantation, for human organs during for transplantation whilst they're being transported. This lamb here contains a peptide called calcitonin, which hopefully would help prevent the onset of osteoporosis or loss of calcium in bones as people age. Preventing human disease. This is an animal called, this is Annie, and she's resistant to mastitis. She was made at the USDA in America. So that is of beneficial benefit to the farmer, an agricultural benefit. This animal didn't get staphylococcal mastitis. Another one is interesting. This is transgenesis of food and the environment. This is a thing called EnviroPig. Now, pigs are very bad at utilising phosphate in their food. And they're also very bad at polluting the atmosphere by putting a lot of phosphorus in their piggy poo and urine into the atmosphere. These animals have an enzyme called phytase engineered into them, which is in their saliva. So not only do they utilise their food more efficiently, they produce up to 65% less phosphorus in their excreta. So these pigs are a big polluter of the environment, and they're the major animal protein source on the planet. As I said, we also would want to be able to remove, one of the things about genetic modification was to remove genes, and these are just a few examples of genes that people have been working on. This is for research. This is the mad cow disease, or the prion gene. Some people are allergic to milk, cow's milk, so we can alter the composition to make it less allergenic. This is an enzyme which may be involved in putting transplantation in the rejection process. Bovine serum albumin, people would like to remove that, replace it with human serum albumin, so they can take bovine blood effectively and get lots of human serum albumin for treating burns victims. And immunoglobulins, people would like to remove the bovine immunoglobulin locus, replace it with the human one, so we can have cows that make human antibodies. And this would be very good for treating viral diseases, and the American government's very interested, obviously, for germ warfare. And in fact, these cows now exist in the USA. They've been made. And these are just a few pictures that we can do gene knockouts. These are Cupid and Diana. They had their collagen gene removed, which has no phenotype. This was a little lamb that had its prion gene knocked out. But there are lots of other applications in disease models, in studying development, aging, cell differentiation, reproduction. And so there's a whole range, in my opinion, of uses for cloning and transgenics. As I said, with xenotransplantation, the idea would be, and the pig is the favourite because their organs are very similar, we could put cells in culture and we can genetically modify them to remove antigens that maybe cause the rejection if the heart was transplanted into a human. So we can go through the whole process and hopefully produce organs which may not be rejected. And there's been a lot of work going on in this in the USA and also in Korea. Right, one last 5 minutes, just so we can get this subject out into the open for discussion. now that scientists have cloned a sheep, are humans next? I love Time for Kids. This is a great magazine. Anyway, that was a long time ago. Can we clone humans? Well, the answer is probably, although it might be more difficult than in sheep. We don't know because we haven't done enough, you know. But if we did clone a human, would we produce the same person? Well, there's a debate about that. You know, there's this nature-nurture debate, because we're all individuals which arise as a product of our genetic makeup, rather than familial, you know, rather than just being a product of our genes. We're impacted upon by everything else that we were exposed to. But people have suggested that reproductive cloning, or the production of a baby, could be used for a number of things. Revival of deceased persons. Well, if you don't get the same person, what's the point? And do we want to bring everybody that's dead back? Treating infertility, especially for so that people who want a baby but don't want to adopt, and they don't want to have donor eggs or sperm, could have themselves cloned because they want something genetically related to themselves. I personally can't think of anything more horrible than bringing myself up. I mean, I think it would be not much fun. But also, people have suggested that they could do it for non-heterosexual relationships. But there are other things which are coming in which may actually help that one. But one of the problems here, the removal of genetic defects. Some people have actually suggested that we could use the technology to introduce genetic defects. Some people have said, I'm deaf. I want my children to be deaf. So you can ensure my children will be deaf by genetically modifying them. Now, the thing is that this isn't truly cloning. This is using the nuclear transfer technique. Because if you were to use this nuclear transfer technique to remove a genetic defect, you would make a new embryo by normal fertilization. So it would be a unique individual. You could then make some cells from that, fix the genetic defect, make a new embryo, and make one fetus, and then throw it all away. So it's still a unique individual. It's not cloning, but it's somatic cell nuclear transfer. But there are a couple of other cloning-related techniques which are coming into use, which is maternally inherited barriers to development, basically mitochondrial disease and cytoplasmic dysfunction. Now, the mitochondria are organelles within your cells which carry their own genome. And they also are responsible for producing most of the energy in your cells. But you inherit all those mitochondria from your mother in the egg. And so you may inherit bad mitochondria. And mitochondria, a lot of people have mixed populations of mitochondria. And so if you've got if you think you carry mitochondrial disease, or you know you are because it runs in families, then you can either screen out the embryos that have bad mitochondria in them and kill them. But in a lot of cases, it's down to the balance of good versus bad. And they just don't know what the phenotype of the disease will be based on the percentage. So if we could actually use some sort of related technique and get rid of the mitochondria and just put good mitochondria in there, or we could get rid of any other problems in the cytoplasm. So how would we do that? Well, there are two ways. We could we take a donor egg and a bad egg, and we could either swap the spindles over and then fertilize it, or we could fertilize it and swap the pronuclei over. So effectively, what you then end up, although this isn't true, you end up with an embryo which is corrected, but potentially is the product of three parents. It's got genetic material in the nucleus from a mother and a father, and then it's got mitochondria from a third person in the cytoplasm. So the other thing that happened in the 1990s was the isolation of embryonic stem cells from human blastocysts in 1998 by Jeremy Thompson. Now, embryonic stem cells have the propensity to grow in culture forever and to produce all of the cell types in the human body. So therefore, these are the cells which may be useful for a whole range of things